It's very exciting for me to meet all of you today. So in this talk, I will give an update on the current state of the art of the Gastruloids field. And Gastruloids are aggregates of embryonic stem cells that can be used to study mammalian post-implantation development in vitro. And I will mostly talk about our work in mouse gastruloids, but I decided to also add a few slides on the human system that we developed and published very recently. So in mammals, development normally, of course, starts with fertilization. And in the first couple of days after fertilization, the fertilized egg cell divides a number of times to give rise to a multicellular structure that's called the blastocyst, which implants in the uterus at 3.5 or 4.5 days after fertilization in the case of the mouse. And 2 days after implantation, an important process named gastrulation starts. And gastrulation is important because it results in the formation of the mesodermal and endodermal germ layers, which later give rise to most of the organs of the embryo. And gastrulation is also the time point in development where all the body axes are established, and where, for example, the head-tail, anterior, posterior, the dorsal ventral, the back belly, and the left-right asymmetry axes are being set up. And gastrulation is shortly followed by various other important processes, such as organogenesis and somatogenesis. And somatogenesis is the process by which the mesoderm of the embryo condenses into blocks that then later form the muscles and the vertebrae of the embryo. And the processes that direct embryogenesis are traditionally studied using animal models, such as mice, but these come with various very important limitations. It's, for example, very difficult to obtain mouse embryos in large numbers, and that limits their use in high-throughput studies. It's also very time-consuming to generate genetically modified mouse lines, and of course, importantly, results obtained with animal models are not always relevant for human development. The discovery that embryonic stem cells can be derived from embryos and can be cultured in a pluripotent state resulted in solved some of the challenges that I mentioned on the previous slide and allowed scientists to study aspects of development in a high-throughput manner. And while the 2D culture protocols that were developed in the years following discovery of embryonic stem cells are very useful in studying what signals are relevant for what differentiation pathways, these 2D culture protocols do, of course, not capture the complex three-dimensional morphology of embryos. So stem cells can also be aggregated into three-dimensional structures referred to as embryoid bodies. And while these can also while these can be used to study differentiation, they are generally disorganized and they do also not accurately capture the shape of the embryo. And for these reasons, various labs have recently invested a lot of effort in developing embryonic organoids, stem cell-based structures that accurately resemble the morphology of the embryo. And the Gastruloid system is an example of such a model system, and I will focus on the Gastruloid system in this talk because that's what I've been working on. So gastruloids are generated by seeding 300 mouse embryonic stem cells into the wells of a U-bottomed 96 Wells plate. And over a time course of 2 days, these cells sink to the bottom of the well, just simply due to gravity. And once they reach the bottom, they aggregate and they form cluster together to form one spherical, symmetrical aggregate. And when treated with the right culturing conditions, this aggregate then elongates over time and forms a structure that resembles important key aspects of the developing mouse embryo. And after 5 days in culture, these structures actually resemble some aspects of 8.5 days old mouse embryos. And microscopy-based characterizations of this system revealed that they are, for example, able to form a clearly defined anterior-posterior head-tail axis. They can form a node-like structure. They can also form left-right asymmetry, which is important for example, left-right asymmetric development of the heart. They form the intermediate mesoderm, which later gives rise to the gonads and the kidneys in a bilaterally organized fashion. And they are also able to express the Hox genes, which are important for patterning in the correct spatial temporal order. This microscopy-based characterization, however, also revealed that gastruloids are not able to capture all aspects of mouse embryonic development. Gastruloids can, for example, not form a notochord, a mesodermal structure that runs along the midline of the embryo. And importantly, Gastruloids generated with the original protocol that we developed in 2014 during my master's internship. These original gastruloids are also not able to capture embryonic morphogenesis. And this is most clearly seen in the process of somatogenesis. So while in embryos, the mesoderm condenses into blocks, somites, and while these somatic cells are present in more or less the correct location in gastruloids, they do not condense into these blocks that are so characteristic of somatogenesis in embryos. And that basically means that gastruloids are able to capture the raw overall morphology of the embryo, the overall body plan of the embryo, but they are not able, the original gastruloids are not able to form proper morphogenic details that are normally present in embryos. So the gastruloid system had until recently mostly been characterized with microscopy-based technologies, and we thought that it would be interesting to extend this characterization to the single-cell level to characterize them in more detail. In order to do that, we applied single-cell RNA sequencing to 25,000 cells extracted from over 100 mouse gastruloids at the 120-hour time point. At that time point, they resemble mouse embryos at 8.5 days post-fertilization. And this approach revealed the presence of 13 clearly defined cell types in gastruloids. And in order to annotate these cell types, we compared the gene expression levels in these cell types with that of the gene expression pattern in a previously published embryonic single-cell data set. And this approach revealed that most embryonic cell types are present in the gastruloids, although they clearly lack the brain, and they're also not able to form the notochord, and they're also not able to form any extra embryonic cell types. And this is very important because this means that they cannot form a placenta, and that means that they cannot implant in a mother, so they can never give rise to a viable living individual. So this approach for the first time revealed the presence of, for example, endothelial, het mesenchymal, primordial germ cell-like, and NMP-like cells in gastruloids. And NMPs are stem cell-like cells that are localized in the tailbed of the embryo, and they give rise to most of the neural and mesodermal tissues of the trunk of the embryo. And indeed, a pseudotime analysis on our dataset revealed that gastruloids revealed the presence of NMP to neural and NMP to mesodermal differentiation trajectories. And if you follow the mesodermal differentiation trajectory, then you can see that the full somatic differentiation trajectory is present in gastruloids. NMPs differentiate towards pre-somatic mesoderm, then they differentiate towards wavefront. I'll explain this in more detail later. And then these cells differentiate towards early and late somatic fate. And in embryos, the neural and mesodermal differentiation trajectories that we identified are very organized in space. And NMPs are localized in the tailbud of the embryo, in the posterior part of the embryo, while the more differentiated mesodermal and neural tissues, such as the somites, are localized more anteriorly in embryos. And to investigate whether this was also the case in gastruloids, we decided to also apply spatial transcriptomics to gastruloids. And the spatial transcriptomics technology that we decided to use for that is called TomoSeq. In a typical TomoSeq experiment, a gastruloid is sectioned in anterior to posterior direction using a cryotome. And we then collect the sequential sections into the sequential wells of a 96 Wells plate. And after that, we extract the RNA content and sequence the RNA content of these individual wells. And this technology is very useful because it allows us to explore the anterior to posterior gene expression pattern of thousands of genes simultaneously in just one experiment. So in this slide, I'm showing a summary of the most important results that we got from these TomoSeq experiments. I'm showing 6 heat maps. The first 5 heat maps are heat maps from 5 individual gastruloid samples that we sectioned. And in these heat maps, the genes are shown on the y-axis, and the anterior to posterior sections are shown on the x-axis. And yellow coloring means high expression of that particular gene in that particular section, and blue coloring means low expression of that particular gene in that particular section. And in this data set, it's very clear that the gene expression, that the 5 gastruloids that we analyzed are quite similar to each other in terms of their anterior to posterior gene expression pattern. And that is very convenient because that means that we can merge these 5 heat maps computationally into one average heat map that is easier to work with computationally. And then we can, of course, generate a similar data set for mouse embryos. So that's what we did. And then we can also calculate an average mouse embryo data set. And then we can compare the average gastroloid data set to the average mouse embryo data set. And that actually revealed a lot of new biology about these gastroloids. So here I'm showing 3 heat maps. First heat map is the average heat map of gastroloids generated from a wild-type cell line. The second heat map is a heat map of gastroloids generated, an average heat map of gastroloids generated from an LFNG cell line. And the third heat map is the average embryo heat map. And now if we want to extract biology from these heat maps, then we can do a number of computational tricks. Like we can, for example, cluster the genes that are present in the heat map based on their anterior to posterior gene expression pattern. And if we do that, then we can identify 8 classes of genes that are expressed in 8 specific patterns. And then we can zoom in on these clusters to extract biology from them. So for example, cluster 1 looks interesting because cluster 1 contains genes that are clearly localized in the anterior part of the embryo. And these genes are localized in a very messy, disorganized pattern in gastroloids. And they're actually also very lowly expressed in gastroloids. You cannot see that in these heat maps because the heat maps are normalized. But we know that from looking at the raw data. And then if we zoom in on this cluster, then we can see that this cluster contains loads of genes that specifically mark the embryonic brain. And it does make sense that we detect it anteriorly in embryos and that it's not detected in gastroloids. Makes sense because gastroloids do not form a brain. Then we also detect somite, a cluster that contains somatic genes, and these genes are expressed in a more central location in embryos, while they are more anteriorly localized in embryos. And this is linked to the absence of the brain in gastroloids. And we also detect, for example, a heart-like cluster. And if we zoom in on individual genes in this heart-like cluster, then we can see that there is a heart-like structure present in the anterior region of gastroloids, but it's not very clearly defined. So this heart-like structure in gastroloids does, for example, not express all markers that you would express, that you would expect to be present in the embryonic heart. So gastroloids are able to form a heart, but it's not really formed properly in gastroloids that are generated with the original protocol, indicating that there is still quite some space for optimization. And we can also do one other computational trick that I just wanted to share with you, and that is that we can also combine this spatial transcriptomics data set with single-cell RNA sequencing data. And that revealed that gastroloids, for example, have a somite differentiation front, which is located in the center of the gastroloid. And the somite differentiation front is very interesting because this differentiation front is in embryos very dynamic. So what happens in embryos is that this differentiation front moves in posterior direction over time. And while it moves in posterior direction, it lays down the somites in its wake. So it induces the embryonic mesoderm to differentiate towards a somatic fate. And based on our TomoSeq data, we could see that this differentiation front was present in gastroloids, and we decided to follow up on that. And first of all, we wanted to confirm whether this differentiation front is indeed present in gastroloids. And to do that, we made gastroloids from an LFNG reporter line. And LFNG marks the differentiation front. And the images of the gastroloids made from this reporter line indeed very clearly showed that gastroloids do have such a differentiation front. And when we then performed live imaging experiments, we could see, and we were very, very excited about that, that this differentiation front is indeed dynamic in gastroloids and moves posterior over time, like it does in embryos. And this is something that we cannot see in our TomoSeq data because TomoSeq is always done on fixed samples. A second important component of somatogenesis entails the somatogenesis clock. So in embryos, there are waves of wind and notch signaling that travel from the posterior part of the tip of a gastroloid towards the differentiation front with a periodicity of 2 hours. And these waves also express LFNG, albeit at very low levels, so it's a bit difficult to see in our movies. But if we zoom in on the movies and if we measure the intensity of the LFNG signal in the pre-somatic mesoderm of the gastroloid, then we can actually see that it oscillates with a periodicity of 2 hours. And this is exciting because that means that the somatogenesis clock is active in gastroloids with dynamics that are very similar to the dynamics in the embryo. These periodic oscillations are thought to regulate the periodic formation of new somites that appear in anterior to posterior direction with a periodicity of 2 hours. So a new pair of somites is being added every 2 hours in embryos over time. And during these live imaging experiments, we observed something unexpected and very exciting. Because during these live imaging experiments, we had to embed gastroloids in matrigel to stabilize them, because gastroloids were continuously drifting away from the microscope, from the field of view, and that was very annoying. And we used matrigel to stabilize them. And unexpectedly, that resulted in the appearance of indentations in a small percentage of the gastroloids. And it might be a little bit easier to see in screenshots that I made from this movie. So you can see that there is, in some of the gastroloids, there are very clear indentations, and these appear right after the somite differentiation front. And we got very excited about that, because we immediately thought that these indentations could perhaps represent somites, and no one had ever before been able to make somites in a dish. So these data suggested that matrigel embedding induces the formation of somite-like structures in this in vitro stem cell-based embryo model. And to follow up on this, to test this hypothesis, to test whether we indeed made somites, we stained the gastroloids for UNC-X 4.1. And UNC-X marks the posterior halves of somites in embryos and is therefore expressed in a stripy pattern in embryos. And when we then stained matrigel embedded gastroloids for UNC-X, we observed such a stripy pattern in 1% of the gastroloids, suggesting that matrigel embedding can induce the formation of somite-like structures, but only in 1% of the gastroloids when you embed them in pure matrigel. But based on these observations, we started to play around with the protocol and we started to optimize the protocol. And in the end, we figured out that if we decrease the concentration of matrigel, we dilute the matrigel, that then we can actually induce the formation of proper somite-like structures in these gastroloids. So we found that if we add 10% matrigel to gastroloids at 96 hours after aggregation, that then induces the formation of very clearly defined somite-like structures in this in vitro model. And if you zoom in on these somite-like structures, you can see that they even have very clearly defined anterior and posterior halves. So this is one somite, which will later give rise to one vertebrae of the one rib of the embryo. That's a bit of a, and in embryos, these somites have very clearly defined anterior and posterior halves that have different functions. And if we zoom in on these somites, you can see that the gastroloid somites indeed also have clearly defined anterior-posterior patterning. And I could actually also confirm that when we performed a double staining on UNC-X and TBX18, which mark the posterior and anterior halves of these somites, respectively. And live imaging experiments then also revealed that these somite-like structures appear with a periodicity of 2 hours. So there's a new indentation, a new block being formed every 2 hours as the gastroloid grows over time and develops in a dish. So the timing of gastroloid development is similar to the timing of the development of the in vivo embryo. And now before moving to the human part of the talk, I wanted to just quickly highlight that we are not the only ones that are working on optimizing the mouse gastroloids protocol. There was a group in Berlin who independently of us also found that matrigel embedding induces the formation of somite-like structures, and thus results in the formation of embryo-like structures that more accurately resemble the actual mouse embryo. And there's also various other labs that are optimizing various other parts of the gastroloids protocol. And Matthias Lütolf's lab in Switzerland, for example, recently managed to improve the formation of the heart-like structure in gastroloids, and they found that adding cardiogenic factors in an extended gastroloid protocol results in a reproducible formation of a beating heart-like structure that also folds over time in a way that is similar to how the embryonic heart folds and shapes itself over time. So then I want to switch to the human part. So we scientists, we know very little about human development. And the reason for that is that it is very, very difficult to study gastrulation in human embryos for both technical and legal reasons. It is, for example, in most jurisdictions not allowed to study live human embryos, to keep human embryos alive for more than 14 days in a dish after in vitro fertilization, while gastrulation only starts at day 60 in humans. So we know very, very little about gastrulation in humans, and we therefore thought that it would be very exciting and interesting if we could generate gastruloids from human embryonic stem cells, because that would allow us to study human gastrulation in a 3D context. And after a lot of trial and error and a lot of optimization, our collaborators in Cambridge managed to obtain elongating structures from human embryonic stem cells. And microscopy-based characterizations revealed that these human gastruloids do also form all the 3 germ layers that are normally present in embryos, similar to the mouse gastruloid system. So they form both the ectoderm, marked by SOX2 in green, and the endoderm. So this is the tissue layer that later forms the gut. And they also form the mesoderm, which forms the somites amongst other tissues. And this mesoderm is marked in blue in these images and in this movie. And additional stainings revealed that this first version of the human gastruloids protocol is also able to generate a proper anterior-posterior axis, and they are also able to form a heart-like structure, although it's not formed reproducibly yet in this first version of the human system, which we will probably optimize in the coming years. And we can keep these gastruloids alive for 3, max 4 days in a dish, and after that they fold onto themselves and they collapse. And we don't know yet why that is happening. And a TomoSeq-based comparison between human and mouse gastruloids and between human gastruloids and samples from the Carnegie Atlas of Human Development revealed that these human gastruloids recapitulate human embryos between more or less 18 to 21 days after fertilization. And that means that we can now, for the first time, start to study human gastrulation in this stage of development that was previously not accessible for research. And it's important to highlight that these human gastruloids do also not form any anterior neural brain structures. And they're also not able to form any extra embryonic tissues. And it was partly for these reasons that these experiments were approved by ethics committees that were consulted before we performed any of these experiments. And it is also important to note that the first human version of the first version of the human gastruloid system was published only very recently. And therefore, the human version of the system is still very preliminary. So it's not yet as advanced as the currently available versions of the mouse gastruloid system. And we can, for example, not yet managed to induce somite formation in these human gastruloids. We are, of course, hoping to and we are going to try to induce somite formation in human gastruloids in the coming years. So in summary, we applied spatial and single-cell transcriptomics to the original version of the gastruloids protocol, which revealed that most embryonic cell types are present in gastruloids and that they're also present in the correct anterior to posterior location. We followed up on these findings with live imaging experiments that revealed that the somatogenesis clock is active in gastruloids. And based on these, and then we also managed to optimize the mouse gastruloids protocol, resulting in the formation of somite-like structures and resulting in general in gastruloids that much more accurately resemble the developing mouse embryo. And we developed the first human version of this system, which is not yet able to form somite-like structures. And one last slide on the applications of this field. So one main advantage of this gastruloid system is that it is relatively easy to generate gastruloids in large numbers. And that makes the system compatible with screening procedures. And we can now, for example, start to use this system for toxicological screenings to identify drugs that identify components that can induce congenital malformations, and that should perhaps be avoided during dietary intake of pregnant mothers. We can also use the system probably for genetic screenings to identify new mutations that underlie congenital malformations, and perhaps we can use it to develop and screen for drugs that prevent or reduce the severity of common congenital malformations. And it's important to highlight here that gastrulation is an important time point in development, also for the reason that many congenital malformations actually result from mistakes that happen during gastrulation. I forgot to mention it in the introduction. Another interesting aspect of the system is that we can use it to study embryology from a bottom-up perspective. So we can use the system to, for example, study how the extra-embryonic environment induces the formation impacts embryonic morphogenesis. And it's quite difficult to address such questions using in vivo embryos. And the currently available version of the human Gastruloid system is based on embryonic stem cells, but we hope that we will soon also be able to generate Gastruloids from induced pluripotent stem cells. And if we can do that, then that would allow us to model human congenital malformations in a patient-specific manner. Because if we can, for example, make Gastruloids from iPS cells that are derived from newborns with a heart defect, then we can maybe use the resulting Gastruloids to study this heart defect and to develop drugs that prevent or reduce the severity of this heart defect. And ultimately, but that's a bit more long-term, ultimately we hope that the system can also be used as a source for tissues that can be useful in transplantation purposes in clinical settings. Okay, and with that, I would like to thank all the many people that have been involved in this project. So first of all, Alexander van Oudenaarden, my PhD supervisor, and of course Anna Alemani, who did most of the data analysis of this project, and Vincent van Batenburg, who helped a lot with the microscopy data that I showed. I'm very happy that I was able to collaborate with Alfonso Martinez Arias and Naomi Morris and others in Cambridge. And we also had a lot of help from Catharina Sonne, and somatogenesis expert and new PI in the Hubrecht. So thank you all for listening, and I'm looking forward to take any questions that you may have on our findings.
Right, good evening. So my name is Jeffrey Aalders, and today I will talk about my PhD project, which is about the modeling of Marfan cardiomyopathy in vitro using stem cells. So first, a little bit of background about Marfan syndrome. So Marfan syndrome is caused by pathogenic variants in fibrillin-1 gene. There are currently more than 3,000 different pathogenic variants described in the fibrillin-1 gene. The genotype-phenotype correlations are very difficult to predict, so it's very difficult syndrome to model. Then the fibrillin-1 gene is encoding for fibrillin-1 protein, and this is a very important protein in the extracellular matrix of the cells. So Marfan syndrome patients suffering from Marfan, they have systemic abnormalities, but most severe is the involvement of the cardiovascular system, and in particular the aorta. However, in a subset of Marfan syndrome patients, sudden cardiac death is observed. Also, there's an accumulating amount of evidence in the literature that there is also cardiomyopathy associated with Marfan syndrome. But the question remains, is this intrinsic cardiomyopathy, or is this cardiomyopathy secondary to the abnormalities in the vascular system? And this is why in my PhD project that I'm using stem cells. Stem cells have different benefits. You can obtain large numbers of study material, and of course, as you can understand, if you want to model something cardiac, it's very difficult to obtain large amounts of cardiac material from patients. So I want to briefly show the workflow in our lab. So we start from urine. We collect the epithelial cells from this urine sample. This is cells that are shed from the bladder. This is how it looks like in culture. And then we can expose these epithelial cells to a Sendai virus. The benefit of a Sendai virus is that it's not integrating in the genome, but it's causing an upregulation of the pluripotency genes. So if you do so, then this picture is after 7 days of the start of the exposure to the Sendai virus. Then we see dense patches popping up. So this is a very early transitioning of the epithelial cells to the induced pluripotent stem cells. And if you then continue, on this slide you can see that we now have established several Marfan syndrome iPSC lines. Of course, we have to do many characterizations to make sure that everything is correct. And one of those characterizations is looking for if they are still pluripotent. So in this slide, I show a NANOC staining, and NANOC is a pluripotency marker indicating that these lines are pluripotent stem cell lines. So to recapitulate this part so far, so our strategy is to start from the patient, so to collect healthy or Marfan patient materials that are somatic cells. In our case, we choose to use urine. Other groups are also using skin biopsies or blood samples, for instance. We reprogram them to pluripotent stem cells. And moreover, we can also make use of gene editing tools such as CRISPR-Cas9, where we can specifically repair this pathogenic variant in the fibrillin-1 gene, creating an isogenic line. And the benefit of an isogenic line is that the genetic background is the same. So while establishing these iPSC lines here in Ghent, we also started a collaboration with the University of Cambridge, where 2 lines were already established. So they had a corrected Marfan line and a Marfan iPSC lines. And with these lines, we started differentiating them to cardiomyocytes. So we optimized different protocols to obtain cardiomyocytes. We are now mainly using a small molecule method. And on the left, I'm hoping that the videos are working, so I hope the internet connection will be sufficient. We see this beating heart cells in a dish, and this is actually forming the starting material for our analysis. So last year, we were able to make a first publication with about the first in vitro cardiac model for Marfan syndrome, and we did many analyses, and I will show a couple of the functional characterizations that we performed. Being the atomic force microscopy, the multi-electrode array, the flex cell, and the muscle motion analysis. So, multi-electrode array to start with. This is a technique that you can compare with an in vitro ECG measurement. And how it works is that we culture cardiomyocytes on top of a grid of electrodes. And we can then measure the extracellular field potential. And from there, we can then derive things like parameters like the beat rate, but also arrhythmogenic events. Then another tool that we used was the flex cell mechanical stretching tool. This is actually a flexible membrane on top on which you can culture cells. And by using a vacuum, you can stretch these cells. And in our case, we can then mimic the hemodynamic stretch that is normally posed on in the heart. For the 2 other techniques, I will go a little bit more in detail. So first, the atomic force microscopy. So what is this? This is actually a small needle that goes over the surface of the cells, and it measures the height as well as the stiffness at every area. And by doing so, we generate then a force map and a height map. And by combining these 2 data sets, we were able to calculate the stiffness of the heart cells. And if you compare then the Marfan cardiomyocytes, which are in red, with the corrected cardiomyocytes, we see that the Marfan cardiomyocytes are very much stiffer. So also here, I'm hoping that the videos are working. And if they're working, you see beating left and here on the right with the Marfan syndrome. And what was striking to us was that the beating was much more pronounced in the corrected. So hopefully you can also appreciate that. And there's different ways that you can analyze this kind of videos. So we use the muscle motion software tool. This is a software tool that makes use of the pixel displacement, and based on this is calculating a contraction amplitude. And if we apply this software tool on these videos, we can actually see that the contraction amplitude is much larger in the corrected compared to the Marfan. If you do then the statistics, we can also see that is a very significant difference. And of course, we can then possibly we can relate back these results with the findings with atomic force microscopy, where we showed that the Marfan cardiomyocytes were much stiffer. Yeah, but those results that we published were all done in 2D, and we were able to show differences between the two different cardiomyocytes. But this also raises questions about what are the mechanisms behind these abnormalities. And to understand better these mechanisms, we initiated co-culturing. We know that cardiomyocyte and cardiac fibroblast are important residents of the heart. And we know that they are both producing matrix and they are both producing fibrillin 1. So as we know, that if there's a pathogenic variant present, it will create impaired matrix. And then in turn, the impaired matrix might affect the behavior and the function of these cell types. And now the advantage of doing in vitro research is, of course, that we can combine different cell types of different origins to see what their contribution is to the phenotype. So what we are now conducting in the coming months is that we combine, for instance, healthy cardiomyocytes with cardiac fibroblasts from Marfan, and the other way around, to see if there's any compensation in their phenotype. And if so, then we can pinpoint more what the mechanism is behind the phenotypes. As mentioned previously, everything is done in 2D. Co-culturing can be performed as well in 2D, but we also want to extend now into 3D. So that's why we last year published a proof of concept paper using liquid marble technology. This is a technology that makes use of a hydrophobic powder. In our case, that is a fumed silica. And how it works is you have a cell suspension. So here we started with a cell suspension. This can be the cardiomyocytes, but of course this can also be a co-culture. We place a little droplet in a hydrophobic bed. We swirl it around, and then we see that the hydrophobic powders, the particles, will encapsulate the marble, creating a microbioreactor. And by creating, it's creating the opportunity to create cardiospheres. So we can also then transfer these liquid marbles into smaller petri dishes. We can incubate them in an incubator, and in a relatively short time period, only 24 hours are necessary, these cardiospheres are formed. Then these cardiospheres look like this under the microscope. So this is within a liquid marble. You see several cardiospheres. And then if you do a staining for troponin T, which is a cardiac marker, we can see this nice cardiosphere. Then many of our downstream analyses are performed in 2D. So what we also do with these cardiospheres is that we plate them. This is after 24 hours of plating. This is after 48 hours of plating. And then what we can do is we can stain them. And so this is very preliminary data, so it's still work ongoing. So we yet have to confirm it. But what we see is that in the corrected on the left, we see more binucleated cardiomyocytes. And this is an indication of a higher maturation status. While in the Marfan syndrome, we don't see as many. In our previous publication, what we also saw on a gene expression level is what the Marfan cardiomyocytes were initially lacking maturation. So now our hypothesis is that maybe this initial lack of maturation in the Marfan cardiomyocytes might explain the abnormalities that we observe in our in vitro model. Right, I want to thank you for your attention, and if there's any questions, then I'm happy to answer them.
Hello everyone, I'm Robin Demuynck. I'm a PhD student at the Cell-Dev Investigation and Therapy Lab at the Department of Human Structure and Repair. I'm only in my first year now, so just beginning my PhD. But anyways, today I'd like to share with you our work on the 3-delta assay, to quantify different cell death and its type in spheroids. Before we begin, I would like to thank everyone, all of the organizers of this conference, to allow me to present my research here today. Okay. Of course, cell death is an important aspect of cancer treatment. Currently, research is focusing more and more on trying to induce immunogenic cell death or ICD in cancer. This is the process of cells dying in such a way that their death activates the immune system against the cancer cells. While dying, cells will release or show on the surface so-called damage-associated molecular patterns, or DAMPs, as you can see here. These DAMPs are not only able to attract antigen-presenting cells, such as dendritic cells, but also cause their activation to a mature state. These mature dendritic cells can then in turn activate T cells via cross-presentation. Cytotoxic T cells will be formed, which can migrate to the cancer, where they recognize the cancer cells and can destroy them. That is why ICD might be an effective approach for anti-cancer treatment. Our lab and several others have already shown that some cell death modalities can show features of ICD, such as apoptosis and necroptosis. However, the problem with apoptosis and necroptosis is that cancer cells quite easily build a resistance towards these cell death types. That is why there is a great need to study other cell death modalities for their immunogenic potential and to use this knowledge for anti-cancer approaches. In our work, we focused on ferroptosis, a cell death type discovered in 2012 by Professor Stockwell and his colleagues. Ferroptosis is characterized by oxidation of lipids and phospholipids in the membrane, which leads to membrane blabbing and eventually to cell death. The mechanism of ferroptosis has already been thoroughly studied, as you can see here. In our work, we induce ferroptosis using either RSL3 or ML162, both of which block GPX4, glutathione peroxidase 4, which is the main enzyme protecting the cells against lipid peroxidation. We decided to focus on ferroptosis since our lab has recently shown the immunogenic potential of early ferroptosis. Early ferroptotic cells were engulfed by dendritic cells and caused their activation and maturation. They also caused anti-tumor protection in mice. All this data suggests that ferroptosis might be an effective approach in anti-cancer immunotherapy. The issue with ferroptosis and cell death in general is that mainly 2D cultures are used for their analysis. 2D cultures are mainly used because they are easy to work with, whereas 3D cultures are more representative for the in vivo situation. But 3D cultures often require sectioning for their analysis or disaggregation into a single cell suspension. These processes are quite labor-intensive and time-consuming. Therefore, we wanted to find an easier assay to analyze cell death in 3D cultures. In our lab, we have a Tican multiplate fluorescence reader at our disposal, and we use this machine as a starting point for our assay development. Of course, before we could develop the assay, we needed to have a 3D model ready at our disposal. We work with tumor spheroids, as you can see here, and we use 3 different cell lines for spheroid formation. The MCA205 and L929 mouse fibrosarcoma cells and the SCOF human ovarian cancer cells were used for seeding on top of non-adhesive aqueous macropores, as you can see here, which allow for cellular aggregation and spheroid formation. As you can see in these graphs, the spheroids remained stable over time, and they became more circular and more compacted over time. These spheroids were then used for the cell death assay. In short, the assay works in the following way. The spheroids are collected and seeded on top of non-adherent multi-well plates. There they are stained with cytox dyes. These dyes are non-fluorescent outside of viable cells, but once cells become permeabilized, like at the end of the cell death process, cytox can bind to the DNA and we can measure an increase in fluorescence. As mentioned before, cell death was induced using either ML162 or RSL3. and also different inhibitors for apoptosis, necroptosis, and pharoptosis were used to determine the specific cell death type that was induced. At the end of the assay, all cells are permeabilized using Triton X100, a detergent, to determine 100% of cell death. The percentage of cell death is then calculated as the ratio of the fluorescence intensity after the measurement after 24 hours of treatment with the inducer to the fluorescence intensity measured after Triton treatment. Of course, several optimization steps need to be performed for this assay, such as the optimization of the optimal spheroid density per well, the optimization of the cytox concentration, and also the timing and the concentration of Triton X100 to see if all cells could be permeabilized in the spheroid. First, we optimize the cytox concentration. For this, a linear correlation is required between the fluorescence intensity after Triton treatment and the number of spheroids seeded per well. R² is a measure of linear correlation, and this was used to determine the optimal concentration. As you can see for this cell line, 3.3 micromolar had the highest linear correlation, and this was then used in all following experiments with this cell line. Of course, this will be different for all different cell types used in the assay. So when you use a new cell line in this assay, you will need to optimize the cytox concentration. And it can also differ between 2D cultures and 3D cultures. Here we also determined the optimal spheroid density, where we saw the steepest increase in fluorescence, which was for us 80 spheroids per well. Next, we optimized the gain settings. Here we also need a linear correlation between the fluorescence intensity after Triton treatment and the number of spheroids per well. What you can also see here is that 2 hours of treatment with 0.05% of Triton X100 was sufficient to permeabilize all cells in the spheroid. When comparing normal spheroids with trypsinized disaggregated spheroids, so a single cell suspension, we observed no statistically significant differences in fluorescence intensity, meaning that for this assay, we can use intact spheroids for their analysis. After these optimization steps, we use the 3-delta assay for pharoptosis induction in spheroids. We compared both day 1-old spheroids with day 10-old spheroids and with the 2D culture of the same L929 mouse fibrosarcoma cells. As you can see, pharoptosis was specifically induced since only the 3 pharoptosis inhibitors could reduce cell death, whereas the apoptosis inhibitor ZVAT and the necroptosis inhibitor necrostatin 1 could not. What you can also see is that day 10 spheroids were a lot more resistant towards cell death induction compared to day 1 spheroids and the 2D culture, presumably because of increased extracellular matrix formation serving as an extra protective barrier. Here, we also compared normal spheroids with disaggregated spheroids. But as you can already see in the control non-induced conditions, cell death was very high, suggesting that this disaggregation process is very harsh for the cells. Also, the data obtained in those disaggregated spheroids could not be correlated towards the 3D models, meaning that this disaggregation process might alter the results as is often required in, for example, flow cytometry. This is the main advantage of this assay, that you can use intact 3D models. Also, you can do real-time kinetic measurements using this machine and using this method. As you can see in this graph, we induced pharoptosis in day 1 MCA 205 spheroids, and we measured fluorescence every hour for 24 hours. So, to sum up the assay, first 3 optimization steps are required, which are to adjust the cytox concentration, to optimize the spheroid density per well, and to determine the best gain settings. Luckily, these 3 experiments can be performed quite fast, and afterwards the assay is ready either for endpoint analysis or for kinetic measurements of cell death. So, in conclusion, the 3-delta assay was established in this work. This assay is easily adaptable to other cell death inducers and to other inhibitors of cell death. It also allows for high throughput screening as multi-well plays are used, so you can analyze a lot of different conditions all at once. Besides quantification of cell death, you can also identify the specific cell death type that is being induced during treatment. In this work, it was also shown that 3D cultures are more reliable to the in vivo condition, presumably because there are more barriers present, making them less sensitive to therapy, as often can be seen in vivo. Finally, I want to thank everyone who contributed to this work and thank you all for your attention. If you have any questions, I will be happy to answer them. Thank you.
I'm a starting professor at lead in tissue engineering and biomaterials lab at the University of Ghent. And today I'm going to talk about biosensors for imaging metabolism in mouse intestinal organoids. So one of the main problems probably discussed a little bit here during this symposium is visualization of the organoids and tumor spheroids and also gastruloids indeed. And for instance, the stem cell niche is very important anatomical compartment, which is not only consisting of the stem cells, but also from the topographical cues, from physical parts of the extracellular matrix, and trophic factors, and many other parameters, also oxygen. And the problem arises on the level when you ask yourself how you can study the stem cell niche. Is it, no matter if you're approaching it from in vivo models or using the gastruloids or organoids, and what kind of models you can employ? Can you really engineer the stem cell niche? And do you always have to disintegrate or analyze, or you still can analyze it in a live state? And one of the models which we think is very exciting in addressing this question is mouse intestinal organoids and also human intestinal organoids. And this is a very good three-dimensional model of the intestinal epithelium. I would like to thank Arne actually for introducing the term of organoids and correct understanding, correct definition for organoids. So for intestinal organoids, we see it as a nice example of really archetypical organoid, where you have a multicellular three-dimensional entity which is sustainable, which grows in culture for many years, and it displays presence of all the functional types of the intestinal epithelium. It's three-dimensional, and what is also important about the intestinal organoids is that they can be grown very quickly. So from the single cells, from using IPS, for instance, you can produce them within 10 days as a functional units, as a mini organs. What is also important that you cannot grow them without hydrogel and the matrix containing basal membranes, and you have to grow them most likely in the matrigel matrix. And one of the approaches which we think is very important and very useful for approaching the complexity of the intestinal organoids and different other related models is imaging. And we think it's because you can also do single cell analysis and minimize cost of culturing. So you don't have to grow hundreds of organoids to analyze their physiology. You can grow just dozens of them and use it as experimental points. And it can also give you mechanistic insight at the molecular and subcellular levels. And you also can address challenges of organoid tissue engineering by using imaging. And we believe that this is very nice emerging alternative to, we think, destructive methods such as next generation sequencing and other ones, or at least it's complementing them. And the tool that we are going to use is actually called fluorescence, FLIM, and phosphorescence FLIM lifetime imaging microscopies. This is very elegant physical methodology which enables you to measure the state when you excite any fluorescent dye. Before it emits the light, it spends some time in the excited state. And if you have the equipment such as a FLIM microscope, which enable you to measure this luminescence lifetime, then you can actually probe many parameters and biomarkers such as molecular oxygen and hypoxia in real time. You can label cell proliferation. You can also study protein interactions using FLIM FRET approaches and many other parameters. So here they're presented as kind of gift boxes. And what is very important about luminescence lifetime imaging is that it enables you to do live cell optical imaging, and it extends the palette of multiplexed fluorophores. So you can also, not only use red, green, blue dye, but you can also add more dimensions based on the luminescence lifetime. Okay. And when it comes to the measurement of organoids, spheroids, gastruloids, or any physiological, the engineered model, the kind of question arises on how you can design the biosensor and how you can deliver it into the, the model you're going to use. And if we talk about organoids or spheroids, which grow in matrigel, so normally, you have to face a problem of delivery of the biosensor through the layer of the matrigel, and basically, especially if you want to approach it in live state, or if you have how you can deliver the biosensor into the cells. So there are different approaches. So you actually, in case of organoids, which have epithelial monolayer, and they have also lumen. And basically, one of the approaches is to stain epithelium using cell staining probes. We're going to talk about it in the first story. Another way is actually to label the lumen. In lumen of organoid, you can also introduce microbiota, as was shown during the last year. And you can co-culture organoids with the physiologically important microbes. So you can stain bacteria, for instance, and deliver them into the organoid. And in another case, you actually can label the matrix in which organoids do grow. And by using, for example, fluorescence markers, you can understand without staining the organoid what happens to proximity of the organoid. And we're going to talk about it in the second story. I'm not going to talk today about injecting bacteria, but we're also thinking about this. So probably during the next presentation. So the stories for tonight, first of all, I'm going to give you an example of how you can image organoid oxygenation and the energy production pathways using the FLIM biosensors. And the second story will be about combining the biosensor scaffold materials, so labeling matrigel, with the analysis of dynamics of lipid droplets. So speaking about the analysis of oxygenation of organoids, so we worked during my postdoctoral years, we worked on the designing different phosphorescent oxygen sensing probes. And for instance, one of them we designed and published already 7 years ago. It's based on the metalloporphyrin, can be excited with the blue or orange light, and it emits in the really red 650 nanometer range. And it shows very useful range of phosphorescence lifetimes. And basically what happens to the phosphorescence or fluorescence, if you measure it as fluorescence, is that it changes and depends on the oxygen. And if you're able to measure phosphorescence lifetimes, you can calculate phosphorescence decays, and you can set up a Stern-Follmer calibration, and you can actually transpose fluorescence lifetimes or phosphorescence lifetimes in this case into the oxygen concentration. And a few years ago, already nearly 4 or 5 years ago, we actually decided to apply this phosphorescent oxygen biosensor to label organoids produced from the primary adult stem cells from the mouse intestine. And we labeled, and what we found actually that organoids displayed very nice and active deoxygenation. So when you expose them grow them at 20% of oxygen, standard conditions, we found that organoids already experienced in the cells something like 10% of oxygen. And they also displayed presence of the gradients. And if you know about the intestinal organoids, they are consisting of the villi and crypts. And different organoids, they can have different numbers of villi and crypts per organoid. And we really seen a very strong heterogeneity of the organoid oxygenation. So between different organoids, just growing next to each other, we had very diverse set of oxygenation data, and we thought, okay, this is very strange, and this is really interesting problem. And we published in actually 2017. But then after that, we decided to look deeper if this heterogeneity, is it really a problem of tissue engineering, or is it having any kind of functional meaning? And we looked at the other approaches based on the FLIM, indeed, which can enable you to analyze mitochondrial function and cell metabolism of organoids. And we looked at also autofluorescence NADH FLIM. And we looked at these hypoxia oxygen probes presented before. There is also another story, which I'm not going to tell you today, but we can also measure mitochondrial polarization in the organoids using FLIM. And so how we did it, we took this LGR5 GFP organoids. So first described by Hans Klever's group from Utrecht University. And what these organoids display is that they express, they are stably expressing LGR5 GFP in the stem cells. And when stem cells do differentiate, they lose expression of GFP. So what we did, we actually looked at the organoids which can be grown in the conditions promoting embryonic conditions. For example, if you have VNT or ENR-VC media, or if you withdraw VNT, they start to differentiate. And we actually labeled them with this oxygen probe, and we looked at the oxygenation in the regions of organoids which correspond to the stem cells enriched areas and non-stem cells. And what we found also, we looked at the media composition. So with organoids, normally people use commercially defined media, commercially described media, and they actually enjoy presence of a lot of glucose. So the standard concentration is up to 20 millimolar, which is very, very much in our opinion. And what we decided to do, we decreased slightly amount of glucose, and we also changed organoids to the glucose kind of deficient media, where we found that we could decrease glucose content to 0.5 millimolar. If we used lower glucose concentration, organoids did die, but they were okay with 0.5 millimolar. So what we did then we looked at the oxygenation in regions enriched with the stem cells and in regions without stem cells. And we found that just simple exposure of organoids to the high glucose medium and exposure to low glucose medium for only 2 hours led to the changes in the oxygenation between these regions. So GFP plus and GFP minus, and the difference was becoming more statistically significant. So we thought that this probably implies that when you deplete median glucose, organoids start to utilize oxidative phosphorylation more efficiently to match their energy requirements, right? So, and now why we actually needed another approach. So we took this NADH FLIM methodology, where using 2-photon FLIM microscope, you can analyze endogenous presence of NADPH, endogenous metabolites, and based on the fluorescence lifetime of NADPH, you can distinguish between fraction of the glycolytically involved and OXPHOS involved NADPH. So in this experiment, we also had these LGR5 GFP organoids, but without staining them additionally, we just analyzed NADPH autofluorescence and measured fluorescence lifetimes. And what we found was very striking, that in high glucose medium, if you look at this graph, in the stem cells, which are more dark, we have kind of lower contribution of OXPHOS when cells are exposed to the high glucose medium. However, if we just deplete glucose to 0.5 millimoles and incubate for 1.5 hours, we found that stem cells could increase their involvement of OXPHOS very quickly. And as a control, we just exposed organoids to the medium with high glucose, but no pyruvate. So if there is no pyruvate, there is no oxidative phosphorylation for most of the cells. And we found that stem cells quickly decreased their oxidative phosphorylation to lower levels. And what is also remarkable, that differentiated cells, they didn't do anything. They were not able to change their metabolism. So this enabled us actually to draw more or less kind of preliminary scheme of regulation of metabolism in the organoids. And we can see that stem cells, LGR5 GFP enriched cells in the organoids, they have a flexible metabolism, and they can quickly respond by changing their metabolism. They can quickly respond to the available glucose and other nutrients present in the medium. While differentiated cells, which use OXPHOS, and we also believe the enterocytes, they use quite a lot of cytochrome P450 oxygen-dependent consumption. So they cannot really change their metabolism. They cannot start expressing or upregulating this machinery. So this is just the beginning of our story. But I think, do I still have time for second story? Wim?
Sure, go ahead.
Okay. So if you continue to stay with me, so this is very interesting, and it shows how with very simple tools you can access balance of energy production pathways in the complex model of the intestinal organoids. And another story is about what if we just want to label the scaffold instead of extracellular matrix, or scaffold instead of labeling the cells. And this can be done by different approaches. So you can use different dyes, you can use fluorescent proteins which will bind the scaffold matrix, You can use conjugates and dendrimers or nanosensors. And we actually, my team, before joining UHent, we were involved in most of these approaches. And so as a proof of concept work, we decided to design the fluorescent protein shown here on the bottom. Basically, this fluorescent protein is consisting of the scaffold binding domain, for example, cellulose binding domain, and fluorescent protein biosensor. In this case is an enhanced sign fluorescent protein, which shows pH sensitivity, and it changes fluorescence lifetime independence of protonation. And we took this cellulose-based scaffold and grown organoids within the scaffold. And what we did, we also labeled organoids with the oxygen probe, and we measured acidification in presence of in proximity of the organoids. And we found that in principle, you can label the scaffold and you can monitor acidification, which is very important parameter, for example, informing on glycolytic flux or the cell growth. And we could see that the organoids acidification can be probed using this kind of biosensor. So in case of pH sensing protein, we didn't really have to label organoid. Okay, and then we decided to move to more physiological matrices. We looked at the collagen matrices, and we took matrigel. We looked at the matrigel, and what we did, we found this collagen binding peptides, and there is a lot of collagen binding domains and polypeptides known with amino acid sequences up to 50 amino acids. And we took the short one called VWF, von Willebrand factor peptide. And what we did, we genetically fused it with the fluorescent protein FRET biosensor, which senses calcium. And it shows calcium sensitivity. It has low affinity to calcium, so it can measure calcium changes in range from 0 to 5 millimolar concentrations, which are matching to the extracellular calcium in the medium, for example. And so what we did, we fused it, we produced it by bacterial expression, and we tested if this kind of fluorescent protein called BD Twitch can label matrices, and we just took collagen 1, collagen 4, and matrigel matrices. And then we also tested if we can use it in the multiparametric FLIM. So what if we have LGR5 GFP organoids and we have a matrix here? So this diffuse staining is actually fluorescence of the matrix, and the acceptor and the donor FLIM fluorescence, and we can also combine it with labeling of other parameters, for example, mitochondrial polarization. So in the experiment I wanted to show you here is that we looked at we decided to perform proof of concept work with this calcium sensor and see if we can monitor extracellular calcium, basal membrane associated calcium, free calcium dynamics in correlation with the metabolism with actually lipid droplets. And we took this dye called Nile Red, which also shows changes in fluorescence lifetime and which can label organoids nicely. So shown here, so it labels different populations of the organoids, and based on its hydrophobicity of what it binds, you can see very different colors, and actually colors correspond to different lifetimes. And what we did, we had organoids, labeled them with labeled matrix with the collagen binding domain having extracellular calcium sensor. We labeled lipid droplets in the organoid, and we exposed them to the chelation of calcium by simply adding AGTA. And what we found is normally, yes, you add AGTA, you experience this decrease of calcium, and you indeed see the decrease of calcium measured. However, what we found was, I think it's shown here in the fluorescence lifetimes, we found that when we exposed chelated calcium very mildly, we saw that organoids were also trying to compensate the extracellular calcium. And we also saw it in response to the changes in the lipid droplet composition. So this is very interesting data. I think probably it's very nice proof of concept work showing potential significance of correlation between lipid droplet metabolism and calcium. And on the other hand, I think we're going to use this collagen binding domain for designing pH and glucose biosensors, sensing pools of these analytes in proximity of the organoids. So the future research plans for our group are that we're going to design new biosensor scaffolds, move towards multi-parameter 3-dimensional FLIM of complex intestinal organoids, and we're also trying to start new co-culture experiments with E. coli and visualize the interactions, metabolic interactions of organoids, epithelial cells, and E. coli. And here I would like to thank my collaborators, my team, current team at UHand, former colleagues and collaborators, and yeah, starting grant both. And thank you for your attention, and yeah, I'll be happy to take your questions.